And welcome to the first lab for Hobbs High School, the biology department. This is Mr. B. We've changed schools yet again. These will be up on the science page under Hobbs High School. But our first lab is the unique properties of water. In this lab, we're going to be examining some of the different things that water can and can't do and showing you some of the different properties that water has. We'll also be using milk because the white, creamy texture of milk is going to illustrate some of the fascinating properties of water a little bit better than if we just did water by itself. So first, a little bit of background on what milk is. Now, this information came from Wikipedia, and before you start yelling, I did confirm the sources at Wikipedia. Basically, this first paragraph here is just telling us that human beings started to consume milk about the same time that we started to develop agriculture, about 10,000 years ago. This was a time when human beings stopped being the hunters and the gatherers and started congregating in areas where the rivers would run over their banks in the spring. They could plant seeds. There were fruit trees growing. They could harvest the fruit, harvest their seeds. And at the same time, they started domesticating, and domesticating means that we would bring wild animals and we would tame them down and allow them to live closer to human beings. That's what domesticating means. So, somewhere along the line, some human being decided they were going to try what was coming out of the teat of the mother animal. And lo and behold, they found that it tasted good. We could make different things out of it, cheese and yogurt. Cheese and yogurt, some of the oldest known products made from milk. They've been around for thousands of years. And they also found that it provided some nutritional value to everything. But what is milk exactly? Milk is what is called an emulsion or a colloid. Now, an emulsion means that we have items being put together that normally would not go together in nature. Somehow or other, they're forced together. And a colloid means that there's multiple items being carried in this suspension. In this case, in the case of milk, as we can see here on our chart, let me see if I can zoom in just a little bit on that chart there. We have different animals that produce milk: cows, goats, sheep. If you live in Asia, it's the water buffalo. The first thing here is how much water's in here. Well, anywhere from 81 up to almost 90% water. Then we have our proteins, our three kinds of fats that are in there, our carbohydrate, which is a simple sugar in the form of lactose, cholesterol, calcium, and then how much energy in the form of kilocalories, or what we would normally just call calories, is this. And of course, asking the students in class the other day, we found out that there were 5 kinds of cow milk available in our local supermarkets. You've got your whole fat, which is 3.9% or 4% fat. You've got your 2%, which of course is half of this, your 1%, which is even less than this, your non-fat or your skim milk, which is 0% fat, and then my personal favorite is the one where they've taken the lactose out of the milk. It seems to make my chocolate milk sweeter for some reason. Going back to lactose, we have some human beings that are what we call lactose intolerant. That means that their body has not developed the ability to process this simple sugar. And when you think about the whole era of human history going back some 2 million years or so, we've had 2 million years to develop the ability to process the sugars out of plants, sucrose, and the sugars out of fruits, fructose. And it's only been in the last 10,000 years or so that we've been drinking milk on a regular basis. So some human beings, their genetic code is not caught up with the program yet, and they're still not able to process that. People that are lactose intolerant, they can have stomach aches, they can be bloaty, gassy, they can have diarrhea. It's just not a pretty picture, and we shouldn't tease anybody that's lactose intolerant. They can't control it. So, our first question on here, if you're following along with your own lab form, is the essential question is, what is an emulsion or a colloid? Well, an emulsion is 2, and I'm going to use the number, it just makes it go faster. I realize proper English, we write out the numbers from 0 to 10 and then we use the numbers over that. And pardon me for sounding hoarse, I'm just battling allergies and just did not want to come in and do this today, but here I am. So there. Emulsion is 2 or more items, substances, put together, combined, compounded, mixed, whatever you want to call it, together, which normally do not. Okay, so that's A. Now the second one is an emulsion similar to a mixture. And it is, yes, it is a mixture. It is similar. Now normally things that are put in emulsions like milk don't normally separate, although we know that milk over time will start to separate. And it will separate into its component parts, especially once it starts to spoil. Although fresh milk, those of you that have ever been around fresh milk know, you leave fresh milk that's been freshly harvested from the cow, and you leave it overnight, and by the morning all of the heavier or lighter fat elements have risen to the top in the form of what we call cream. And that can be harvested off, that can be churned into butter, it can be turned into all kinds of different products. And then what we have here is this is just an illustration of some of the different fat globules that go into the emulsion that's in milk. All right, so now we're going to move on into our lab. Excuse me. So we're going to flip the page. We're going to start up here. Remember, when we're doing a lab, we're following the scientific process. We've had some kind of observation or we've identified some issue that we'd like to explore. We're going to come up with a hypothesis. We're going to design an experiment and then perform it. We're going to analyze our data and then we're going to report our results. So for this particular laboratory, the essential questions or observations are A, what happens when different substances are mixed with water? B, is it possible to observe cohesion and adhesion, 2 of our terms from class that we discussed last week and we'll be exploring now because it's very important for water. And then C, how is pH measured and what does it mean? Now we're going to start with C first in this first part of the lab. And let me see if I can get my camera turned around a little bit to go with how I write. So pH is a measure of the acid or base. Now another word for base, we would call this alkaline, A-L-K, alkaline, I-N-E, of a substance. And so this is the first part right here. Now we're actually going to discuss this. We did this in class. I'm going to test. I happen to have here a bottle of Hobbs water fresh out of our water fountain in the hall just across from the door to the classroom. And we're going to say that our Hobbs, H-O-B-B-S, water is blank pH. And then I had students, if they wanted to, I gave them all a piece of pH paper, litmus paper, and had them check and see what their pH was. And we'll do that in just a moment. All right, so pH is a measure of the acid or base, the alkalinity of a substance. Acid is a concentration of what we call hydrogen ions. This is where they're giving off their electrons. And the base or alkalinity is when we have a concentration of hydroxide or OH negative ions. That means they're sucking those in. So in one case we're giving the electrons away, in the other case we're bringing the electrons in. Okay, we use something called pH paper. We have a pH scale as we've discussed in class. 7 right here in the bottom corner is neutral. all of the greens going up to the number 13, and the pH scale actually goes from 0 to 14. But from 8 to 13, this is our alkaline, our base side. And then anything under 7, 6 down to 0, 0 being some of the strongest acids known to man, battery acids down here. I actually brought a Coca-Cola along. We did one in class the other day, and it came in at about a 2 or a 3. But we're just going to start with water first. So I'm going to leave this right here. And I have some of our pH paper that we've previously taken out. And I'm going to take a strip off just like that. And I'm going to open up our bottle of water. Now, hopefully I don't get this all over the table. But I'm going to do this, see if I can do this in front. Yes, there we go. You can see that. I'm going to dip that in the water. I'm going to pull it out. And lo and behold, we are going to test our water, and it's going to come in at approximately 7 on the pH scale. Now, my first two periods, periods 1 and 2 on Friday, we tested the water fresh out of the water fountain, and it actually tested at 8 on the pH scale. And I'm hypothesizing that because the water had sat in the pipes all night, and I'm assuming the city of Hobbs uses a chlorine gas that they bubble through the water to kill the bacteria, and chlorine is on the alkaline side, that caused the water overnight to have a higher concentration of those alkaline atoms in there, the chlorine. And because nobody had gone to the bathroom and nobody had gone to the water fountain, it took a while to get that water through to get it to more of what was actually in the tanks and what's coming out of the ground, which is more of a 7 on our pH scale. And it could be closer, it could be between 7 and 8 now, but it was closer to 7 when we tested it. So that's our pH test there. So our Hobbs water comes in at about 7, which is perfectly A-OK. Now, for me, and I'm not going to show this on camera, but I'm going to take a strip of paper, and I'm going to stick it in my mouth. And the paper does have a taste to it. It's been chemically treated. There, it is what causes the reaction. I'm going to put it in my mouth. I'm going to count 1 and 2. Very good. I'm going to pull it off. And, whoa, definitely a difference right there. Okay, the Hobbs water being this one. Seems to be getting a little darker and on that 8 probably on the pH scale. These are very subtle changes in green, but I'm going to go with more of an 8, so more of a base, but I wrote in 7, that's fine. But I am definitely coming in neutral today at 7. Now, when we tested the students in the different classes, we had everything from 6 all the way up to 8. Students were being concerned, does this mean I'm going to die? And absolutely not. Human beings have a normal range of alkalinity and acidity depending on what you've eaten and drank and you know, your body chemistry. It's perfectly A-OK to do that. So, and then of course also blood is a little bit more on the alkaline side. Urine can go a little more on the acidic side because of what they're doing. You know, your body can have different pHs in different parts. Your stomach acid would probably come in at about 1 or 0 on the acidic scale if we could just squeeze the acid out of your stomach. And what keeps you, and of course there's somebody going, oh my gosh, I've got acid in my stomach, it's going to burn a hole. No, your stomach is lined with mucous membranes that prevent that from happening, and it just is there to help you digest your food. So anyway, I am a 7 on the pH scale. I'm going to adjust that Hobbs one to say 7 and 8. And then as far as we got yesterday was, we got down here to experiment 1. Now our hypothesis for number 1 we have not covered yet. But what happens when different substances are mixed with water? I'm going to go ahead and put this one in, and we're going to go ahead and fill out our hypotheses up here. And the hypotheses up here, what happens when we mix different substances with water? And some will mix, others will not. Very simple, keeping it simple. How is it possible, or is it possible? And we're going to say yes. We will put water on a surface and observe both. Okay, so cohesion and adhesion. All right, down here, experiment 1. What happens when different substances are added to water? I've got a spot here for 3 things, and I'm going to quickly go get them, all the while talking to you in the background. But I've got 3 different substances that I'm going to add to the water, and we will be seeing what happens to our water when I do this. This is what happens when I wasn't quite prepared and then I started making the video, but that's okay. Gives you a chance to pause the tape for a second and get caught up. All right, I've got 3 things that we're going to add to water. One of them, we have food coloring. The other one, I have kosher salt. My wife happened to have that in the cabinet for a while. And then the other thing we're going to add to water is I have vegetable oil. So 3 different substances that we're going to add to the water and see what's going to happen. We're going to start with the food coloring, and this is what we've done already. Let me pull this up and around. This happens to be from first hour on Friday. We dropped 2 drops of red food coloring in. And what you're going to observe in this is going to be diffusion. This is the particles interacting with each other, the food coloring particles and the water particles, and how they're going to exchange electrons, and they're going to swap places, and they're going to find a place, the nooks and the crannies for everybody to hang out. And we're going to get this beautiful color all the way through, even though that didn't happen. Excuse me. This was second hour. We had a nice blue. 4th hour, 3rd hour, we didn't quite get there because we were talking about other stuff. 4th hour, we had another red. And then 5th hour went green yesterday. And actually the green showed us quite a bit of different deformity, I don't want to say deformities, but different properties of the water that was happening. So here we go. Let me get this out of the way right there, and we'll set that down. So we're going to adjust the camera. We're going to set this in front. I'm going to take the lid off the top. We're going to start with the food coloring, and I'm going to go ahead and use the green because it's out, because I think it actually gave some of the better show yesterday on what was happening. So I'm going to drop 2 drops of food coloring in. And here goes drop number 1, and here goes drop number 2. Now we're reusing water bottles to do this experiment, but as you can see already, I've got concentration. I've got a concentration of food coloring towards the bottom. I have some streaks, I have some more dark spots, I have lighter spots, I actually still have clear spots in the water bottle up here and over here. So water, and I think if we get right, oh yes, there it is. You can see the streaks that are going through the water. We normally think of water as pretty homogeneous or pretty consistent all the way through. What this is showing us is that no, water is not homogeneous, it is not consistent, and therefore we need to understand that water, because it's a combination of hydrogen and oxygen atoms, that those hydrogen and oxygen atoms might not be living together in quite the harmony that we think they are. All right, so Let's put this side by side here. So this one from 5th hour yesterday started out almost exactly the same way. Yet this one on the left is what we see today. So what's going to happen over the next 24 hours or so is we're going to see these molecules of food coloring, they're going to start interacting with the water molecules. So you have this concentration on the bottom. It's kind of clear. You can see the streaks through there. But over the next 24 hours or even less, it might even be the next hour or so, we'll check back periodically as I continue to do the lab. But we're going to see this green spread through the entire water bottle. This is going to happen all by itself. It's not anything that's related necessarily to temperature, although this warm room at about 70 degrees will help it along. But this is more just the interaction of the different atoms in here that will get it to this state all by itself. Now, of course, if I shake this, I speed this process along by adding energy and forcing those molecules to go together and change places and knock each other out of the way until they can settle in and everybody can get comfortable over here. This, what you're seeing, this is called diffusion. Diffusion is when you have the different atoms and they're spreading out. This has become a solution. Now remember, a compound in chemistry and biology, a compound is when two or more items are put together. This is a compound, this is a compound. This is undergoing what we call diffusion. This has already undergone diffusion and it has become a solution. A solution is a mixture or a compound that we cannot separate. It is very, very difficult for me to get the green back out of this water. Is it impossible? No. But under normal circumstances, there is no way for me to get the green back out of this water. So therefore, we call this a solution. In a few minutes, when we come back for part 2, we're going to explore what happens when we put the salt and the oil into another bottle of water and see what happens to those two substances when we put them in. So this is going to end part 1. Let me move this out of the way here. So at the end of part 1 of this video, we should have answered our essential question on this side about what is an emulsion or a colloid. On this side, we should have filled in our 3 hypotheses on what we think is going to happen. On C, we answered the question of what pH was. It's a measure of the acid or base alkaline of a substance. We also measured the Hobbs water out of the water fountain. It came in at between 7 and 8. And we also measured my pH, and it came in at a solid 7 on the pH scale. And just a reminder, this is what the 2 strips look like, sitting side by side. I'm definitely 7. This is definitely a little bit more alkaline. And that's probably due to the water treatment that they've done to it. Could also be due to the natural minerals that are dissolved in the water here at Hobbs. When we come back for part 2, we're going to be adding, we're not quite done yet. So substance 1 here, experiment 1. What happens when we add substance 1, sorry about that, was food coloring. And we saw, what did we see? We saw threads. We saw streaks. We saw dark patches. We saw clear patches or areas. Okay, let's just take a quick check of what's going on here. It's darker at the bottom than it is at the top. Darker at bottom than top. And I'm going to go ahead and record the time. It is 1:55 p.m. and then I'll come back and we're going to check this before we're done. So here at the end of part 1, we should have all of this filled out. We'll come back and do substances 2 and 3 and do what happened, and then we will start on the second total experiment when we get done with that. All right, be back in just a second with part 2.